Hello everyone, let's get started. Today we're going to be discussing vaccination. The first thing we need to do is define this important term. A vaccine is defined as a substance that is used to stimulate the production of antibodies and provide immunity. Why do we vaccinate? Well, there are really three different reasons. The first is the most obvious. We want to provide immunity to some particular type of infectious disease to an individual. So we vaccinate that individual to give them that immunity. Number two, we also want to help the community through vaccination. So we want to promote herd immunity in our communities. And we'll be talking about this concept of herd immunity. The third reason for vaccinating you may not be familiar with. We can now vaccinate individuals against certain cancers. We give credit to a man named Edward Jenner for the production of the first widely used vaccine. It was a vaccine against smallpox, and it was developed in 1796. Vaccines have probably been in use much longer than that. There is some evidence that even in ancient societies, people had an understanding that if an individual were exposed to a small amount of an infectious substance, that they could sometimes be protected against the disease that that particular organism caused. But we give Jenner the credit because his was the first widely distributed, historically recorded use of a vaccine. Let's take a couple minutes and talk about this concept of herd immunity. When we are vaccinating in a community, we need somewhere between 60 and 90% of the population in that community to become vaccinated in order to achieve what we refer to as herd immunity. Now, what herd immunity provides for us is three particular advantages. The first one, of course, is that the individuals who are receiving the vaccine are going to be immunized. They're going to be protected against infection. The second reason is not quite as obvious. If we can get 60 to 90% of the population immunized, that's going to remove those people as susceptible hosts for the virus. If there are fewer people who are capable of becoming infected with the virus, there are fewer places where that virus can be replicated. That's going to literally reduce the amount of viral particle that's in that community. If there's less virus circulating, there will also be less disease. That's important because remember, there are going to be people in every community who are not able to be vaccinated. Young infants, the elderly, people who have certain chronic illnesses, and people who are allergic to components that are within vaccines or who have had a previous adverse reaction to a vaccine, those people cannot be vaccinated. By achieving herd immunity within a community, we offer protection to those people who are most vulnerable, those people who cannot be vaccinated, because we are literally removing all the other susceptible hosts, and we are reducing the amount of virus that's circulating. Now this 60 to 90% figure for herd immunity, that number is going to vary depending upon the virus and depending upon how infectious it is, how easy it is to transmit from person to person. You have probably heard that the target for SARS-CoV-2 vaccination is 70%. That is our best estimate for how many people within a community must be immunized against this coronavirus in order for us to achieve this herd immunity. I'm not going to go into great detail about the vaccines that are currently in use to prevent cancer, but there are a couple that you should be familiar with. We currently have 2 vaccines in wide routine use that can help to prevent cancer, and we have one on the market that is used to treat cancer. In terms of the prevention of cancer, we have what's called the HPV vaccine, and that stands for human papillomavirus. And we have the hepatitis B vaccine. The human papillomavirus vaccine protects both males and females against infection with HPV, and that has been shown to prevent the development of cervical cancer in women who get infected with HPV. Up to 90% of all cervical cancers in women are the result of infection with HPV. So you can imagine that it's very important for us to immunize young women and young men against this virus before they become sexually active. The hepatitis B vaccine helps to prevent a particular type of liver cancer that is the result of viral infection. Now, with regards to the treatment of cancer, there is a product on the market called Provenge. Provenge is a brand name. It's not a classic vaccine. It's an actual treatment for cancer using the same theories of vaccination. So this Provenge product allows us to combine the patient's own immune cells that we have harvested from their body with some of the cancer cells from the prostate gland. We combine those 2 cell types in the laboratory, and we allow those immune cells to recognize the cancer cells as being foreign and do what immune cells do, which is develop and produce antibodies against those cancer cells. Those antibodies can then be given back to the patient as a therapy, as a personalized therapy against that particular type of cancer. When it comes to cancer therapy, this type of personalized immune therapy is the most promising advancement we've had in a long time. Next, we need to think about who gets vaccinated. Obviously, infants and children are the first on the list. We begin routine vaccination of infants when they are only a few weeks old, and we continue vaccinations throughout childhood. Teenagers are also vaccinated against certain pathogens that we know they will get exposed to as a young adult. Remember, we vaccinate teenagers, for example, against human papillomavirus. And we also vaccinate against certain infections that tend to occur when teenagers are housed together, for example, in colleges. So there are certain vaccines that are particularly targeted at teenagers and young adults. There are some vaccines that are given specifically to adults, including pregnant women and the elderly. Elderly people begin to lose the strength of their immune system, and so have to have protection against some of the pathogens that tend to hit them as they age. Fourth on the list are what we consider at-risk groups. These are really specialized vaccines that we do not give to the general public. For example, there are vaccines that are given to members of the military that you and I will never receive unless we serve. Things like the anthrax vaccine are reserved for military members because those are the people most likely to be exposed to anthrax as a bioweapon. Healthcare professionals receive vaccines that people out in the general public do not receive, and that's because they are at higher risk of being exposed to particular pathogens at their workplace. Veterinarians are also considered an at-risk group, specifically for rabies. Veterinarians are all routinely vaccinated against the rabies virus. This list is not exhaustive. There are other specialized vaccines for particular groups of people, but these are vaccines that are not given across the larger population. And finally, there are vaccines that are reserved for people who are traveling to certain parts of the world, places where there are pathogens that just aren't found here in the United States. Now, you don't always need vaccinations to travel, but there are certain locations around the world where the State Department will recommend that you get particular vaccinations before you travel. Next, we're going to talk about what a vaccine is actually made out of. There seems to be a lot of confusion in the population about the safety of vaccines, and we will be talking about that in just a few minutes. Part of that hesitance about getting vaccinated comes from the fact that most people don't understand what is inside of a vaccine. It's actually a very basic and short list of ingredients. The first and really most important part of a vaccine is what we would call the causative agent. We have to put something into the vaccine that the immune system is going to recognize and react to. There's certain terminology or vocabulary that goes along with different types of vaccines according to whether we've included the entirety of the causative agent or some part of it. Now, some of this terminology is confusing. Terms like killed and attenuated are often used when discussing vaccines. But as we've discussed in this course, calling a virus killed doesn't really make sense because viruses aren't alive to begin with. But we do use the term to indicate that the viral particle has been somehow destroyed in the laboratory so that only the molecules that made it up are left over and used in the vaccine. That's what's generally meant by this term, killed vaccine. An attenuated vaccine is a little bit different. The other term you'll hear for attenuated is modified live. Again, viral particles aren't alive, but an attenuated viral particle has been left intact, but it has been changed, it has been altered in the laboratory so that it is no longer able to cause disease, to bind to the receptor of the host cell and get inside that host cell. Both killed and attenuated vaccines tend to have the whole organism inside the vaccine. It's just that in a killed vaccine, that whole viral particle has been broken apart. And in an attenuated vaccine, it's left intact, but it's modified. Neither killed nor attenuated vaccines are capable of causing disease. No vaccine is capable of causing the disease that it immunizes against. That's a very, very important thing to take away from this lecture. So sometimes we do put the entirety of the organism into the vaccine with some modifications that have been made in the laboratory. These vaccines are actually hard to produce, however, and we've talked about why they're hard to produce. You have to grow the viral particle in culture, and that means you have to grow a host cell in the laboratory that you can then infect with the virus so that you can get lots and lots of viral particles produced to put into these vaccines. Whole organism vaccines have really fallen out of favor, although we still use them. It's likely that in the future, these whole organism vaccines won't be made anymore. In other words, we won't develop new vaccines that use whole organism technology. The causative agent in some vaccines is actually just a piece of the genetic material that comes from that agent. Now, there are some viruses that contain DNA as their genetic material, so we can harvest that DNA from the viral particles. We can replicate that DNA in the laboratory without having to grow the virus in culture. And then we can use that DNA in the vaccine. Now, there is one big problem with a vaccine that contains DNA, and that's that the immune system does not respond very well to foreign DNA. If you think about it, that's because all DNA is the same. Whether it comes from human cells or from other animals or from plants or from bacteria, DNA is DNA. And it is not a good antigen. It does not stimulate an immune response. So how do we use DNA as a vaccine? Well, the answer is we deliver that DNA into the host cell so that the DNA can be used to make viral protein by the host cell. Host cells will then secrete that protein, and the protein, the viral protein, will stimulate the immune response. Now, there are a couple of important technologies that go into making a DNA vaccine or a vaccine from other types of genetic material. In order to deliver genetic material into a host cell, we can use a viral particle. We can use what's referred to as a viral vector. And we've been using viral vectors for a number of decades now. Generally speaking, what we do is we grow up a virus in the laboratory that we know can easily get entry into human cells, but does not cause any kind of horrible disease in humans. For example, one of the most common vectors that we use for this is an adenovirus. Adenoviruses are known to cause mild respiratory diseases, such as the common cold. We took adenoviruses into the laboratory, again, several decades ago, and we modified them by removing the genetic material. We essentially hollowed out an adenovirus so that it was just a shell. It was just the protein coating and the spikes, the spike proteins that come off of that capsid. In the laboratory, we can take those adenovirus shells, again, what we call the vectors, and we can put DNA of any virus that we want to vaccinate against inside the shell. Now what we have is a delivery device, because the spike proteins on the viral vector, the shell, are going to bind to the human cells and deliver that piece of DNA into the cell. Then the host cell will take that viral DNA, produce viral proteins with it, and those viral proteins will stimulate an immune response. Vector technology is very safe. There is no way that that shell of an adenovirus, that vector, can cause disease inside of us because it doesn't have genetic material anymore. And it works as a beautifully, naturally produced delivery device for other pieces of genetic material. Now, that's the way we would deliver DNA in a vaccine. But as you all know now, having come through the pandemic, our most modern vaccine technology uses RNA. And that's because there are some viruses that use RNA as their genetic material. Remember, all RNA is the same. It doesn't matter if it comes from a virus or from a human or a plant or a bacterium or whatever. If a piece of RNA comes into one of your cells, your cell cannot tell the difference between its own RNA and this foreign RNA. And your cells are going to do what they do with that RNA. Remember, our cells are going to take an RNA and they're going to translate it. They're going to use the RNA to make protein. So by delivering viral RNA into a host cell, we can cause the host cell to translate the RNA and build protein. So if you think about it, a DNA vaccine and an RNA vaccine are sort of similar because the host cells are going to build protein from both of those types of genetic material. The differences are that there's an extra step with DNA. The DNA has to be transcribed first to make an RNA, and then the RNA gets translated. That's the first difference. And the second difference is we can get RNA into a cell without using one of these viral vectors. The vaccines that have been developed against the pandemic coronavirus are RNA vaccine technology. A piece of RNA taken from the SARS-CoV-2 virus can be replicated in the laboratory very rapidly without the need for growing virus in culture. And then that RNA can be delivered in a vaccine without needing a viral vector. The new part of that technology is that the RNA is simply coded. It's coded in a proprietary type of lipid that allows it to go into the cell without needing a viral vector or a viral shell around it. Now you can imagine this particular coding that has been developed in these new RNA vaccines is highly secretive. These pharmaceutical companies are not letting us know what particular type of lipid coating they're using, because that's really the new part of this vaccine technology. That lipid coating around that piece of RNA allows that particle to pass right through the host cell membrane so that the RNA can be translated into viral protein and an immune response can be generated. Some of our SARS-CoV-2 vaccines use this RNA technology. Some of them use the old-fashioned vector technology. It's good that we have a variety of vaccine available against the SARS-CoV-2 virus. Again, we use causative agent in whole or in part in vaccines, but this causative agent is not able to cause disease in the host. It is not possible for a person to develop that disease from a vaccine. The vaccine stimulates the immune system without causing the disease. The second ingredient that we find in a lot of vaccines is preservative. And the reason we add preservative to certain vaccines is because it increases the shelf life of that vaccine. The third ingredient that may or may not be found in vaccines is a diluent. You can see I've put a plus minus in front of diluent because not all vaccine formulations require a diluent. A diluent is simply saline, and it is added in to a freeze-dried preparation of vaccine in order to dilute it, in order to get it into a liquid form that can be injected. If you look at the picture over here on the right-hand side of the slide, you can see a vaccine called Varivax next to a sterile diluent. So the vaccine comes as two containers, a dried formulation of the vaccine that has a much longer shelf life than it would as a liquid, and some sterile saline. We're going to add this diluent into this dried formulation. We're going to mix those two things together, and now we have a fluid that can be injected into the patient. Last on the list is also one of these plus-minus ingredients, and that's adjuvants. Not all vaccines need an adjuvant. What an adjuvant does is provide increased stimulation for the immune system. In other words, an adjuvant is a chemical that all by itself will stimulate the immune system. Some vaccines just don't get a very good immune response from the patient. So we add an adjuvant into the formula to help increase the stimulation of the immune system so we get better production of antibody and better immune memory.